Okay. Hello, everyone, and welcome back to our online class. It's good to see you all again today. I hope Monday's lesson went well for you. And again, reach out if you need anything or have any questions. I'm more than happy to help you. It looks like most of you got it done. So, today we're going to be talking about populations. I think one of the things that's really interesting about populations, I'm going to switch back here, is that they're made up of individuals, but population has a much different set of properties than a collection of individuals do. That's kind of that idea of emergent properties. So we're going to start to consider what populations look like, how populations behave, specifically around age and spatial structure. And we're also going to talk about how populations are measured today. So just move forward with the slides. You'll find these on my courses under the 25th, on March 25th. Strange coincidence. We're talking about chapter 26. And I just wanted to go over some of these characteristics of a population. There's, of course, the size, so how many individuals there are. And one of the things that's interesting is that size can be used to think about what a healthy population is. So the example listed here is the California condors. Population size dropped to 22, but because of this captive breeding program, they've rebounded and they have healthy populations. Something you can go and look up. Density is an interesting idea. That's just how many individuals there are per area. So there's a healthy density of individuals, and then there's an unhealthy density. So thinking about how many we should have fish in a pond is a good place to start thinking about that. The other idea here is the next one is distribution or dispersion. So how evenly distributed throughout the population, or I'm sorry, throughout the environment is the population of individuals, or are they clumped around a particular resource? Often water is something that you'll find individuals clumped around, but there can be other characteristics that may not be immediately apparent, like nutrients in the soil or amount of food that's available to herbivores. Another really interesting characteristics of populations is the sex ratio. Many species have a 50-50, but some have trend towards more females than males. So there's some species of fish that actually just have one male and in the population, and if one of those males dies, one of the females will become a male. That's really cool. In your book, they talk about China and the impact of different policies and how culture comes into play with that. The other interesting characteristic that can be used for understanding populations is age structure. So, how many young individuals, how many old individuals, and what that means for the future of the population. That you want a good balance of young and old so that the population is sustainable. One area where I like doing case studies is around the horses on Assateague Island. You want to look that up. It's a cool little case study. All right. So the last lesson had you thinking about these kinds of ideas around population growth, and so I'll just review them real quick. Right, our selected species have an exponential growth curve. K-selected species have a logistic growth curve. One of the things to understand about the exponential growth curve is that there's no such thing as a population that will grow infinitely. So oftentimes a population that's growing infinitely will experience some sort of collapse, either due to the environment or due to some density dependent factors. And so I just kind of want to move on to that idea of what sorts of things limit population size. They're broken up into these two different characteristics categories. There are density dependent factors, and those are things that have come into play as the population grows. So there's more predation if the size of the prey population gets larger, and that predation pressure will drive down the size of the population, which will make it easier for the population to grow again because there's less competition for food. Disease is another example, that the higher the density of individuals, the quicker a disease will spread and limit the size of the population. Some density independent factors, and the best way to think about this is like, does it matter how many individuals there are? And so something like weather and climate, it, 1,000 deer won't change how much it rains. 10,000 birds won't change how much fire there is, right? Earthquakes, volcanic eruptions, these are all things that are going to happen no matter what. And so those are density independent factors that will limit the size of a population, and those are abiotic factors. All right. The next thing that I want to go over in your activity today will focus on these kinds of things, but just something to point out is that we have 3 different ways that a population can be dispersed within the environment. So you'll see up there the fish. We've got clumped dispersion, so that's something like schooling fish. A lot of plants will also have this kind of dispersion pattern. We've got uniform, so you've got the penguins there that kind of space each other out evenly, but there are other examples out there. And then there's random, so there's no particular pattern to this dispersion, but I think one of the things that's interesting is what can often look random might also be the impact of particular factors, and there might actually be a pattern of dispersion. So you're going to go through an activity today that's going to help you think a little bit about how populations are measured when they exhibit these different patterns of dispersion. So we're going to start with visual estimation, and we're going to move on to quadrat sampling. Quadrat is a very popular technique for studying populations of organisms with ecologists, and I'll talk about that in a little bit, but the gist of it is you just put a square down and you count how many are in that square, and you make sure that you take a random sample of an area so that you get an accurate account of where the individuals are, especially if the individuals are clumped. I'm just going to go back to this real quick. Are clumped or randomly dispersed. You need to make sure that you sample the right area in the right way. Another really interesting way that we're going to work on next Monday is the mark recapture method. So I'm not going to talk too much about this, but this is a way that you can estimate the population of different organisms that move around. And so it's pretty neat because it's pretty simple but powerful method for estimating the size of a population. So I just want to take a minute now to go back to thinking about what this assignment is going to look like, talking about what this assignment is going to look like for you. So I think one of the things that's really interesting to think about is, how do you figure out the size of a population? So if we look over here on the right, or left, I don't even know where it is for you, you have some butterflies. And so if you wanted to figure out how many butterflies there were in the forest, I've got a picture here of the Smoky Mountain National Park. So if you wanted to know how many swallowtail butterflies there were in Smoky Mountain National Park, how would you figure that out, right? The same is true for things like trees. So looking at these hillsides, if you wanted to make a comparison of how many oak trees there were on the right side of the hill versus the north side of the hill versus the south side of the hill, how would you do that, right? And so these methods have been established to support ecological research and coming up with answers to questions like that. So you're going to start with just a visual estimate on this assignment, and you'll find this on my courses under the assignment. I think I got things figured out. Somebody brought to my attention that I had put up 2 assignments. You can take a look and see that 2 assignments for Monday. You can take a look and see that I just did one for this. So you're going to start with a visual estimate of the population size. So if you look at this, how many birds do you think are there? And then you're going to use a grid method to make a better estimate and count up, see how accurate you were. Then you're going to go down to this section here and use some quadrats. So you've got quadrats that are already set up on this diagram. You're going to estimate the population of skunk cabbage, which is, if you're here in the Rochester area or in the Northeast, is coming up now. If you live anywhere near a creek or a swamp, you can go out and probably find some skunk cabbage. And then the other one is jewelweed, which you'll see is a more dispersed clumped population than the skunk cabbage. So you're going to use a quadrat estimate to figure out how many individuals there are in these populations. So follow these directions. One thing that I want to point out is this idea here that your group will receive a list of randomly generated letters and numbers. In the My Courses assignment, so let me just go back here so I can show you. You will see that there is this assignment here, complete it, and then, down here, the 25th. You're going to have to complete it, and you'll notice that I will put in a file that you need to complete this assignment with a random number chart. So basically, you just choose one of these sets of numbers, and these are the quadrats that you are going to sample. So once you've done that, you'll calculate percent error, and you can count up how many individuals there actually were to do a comparison of how accurate your estimates were, or I posted the answer up on my Twitter feed. So if you find me on Twitter, you'll find the answer much easier. So that's what we have for Wednesday. If you have any questions, let me know. But when you finish the assignment, just submit it. And if you have any technology challenges, let me know. I can make adjustments. All right. Well, I hope everything is going well for you. And I hope that you're able to complete this work. And again, reach out if you have any questions. And just a reminder that I'll have the office hours from 12 to 1 on Wednesday. And then Friday we have a literature discussion through Zoom. So make sure that you go on My Courses. You'll find the article, the questions, and a game that we'll play during the literature discussion. And again, if you can't make the literature discussion on Friday, please let me know so I can make alternative arrangements. All right. Thanks a lot. Have a great day.